Dear Professor, how would you rate the clinical efficacy of zanubrutinib in patients with Waldenström macroglobulinemia compared with ibrutinib?
So in the ASPEN-study, I think that zanubrutinib is at less effective than ibrutinib, because in the overall response rate and also the CR and VGPR response rate are at least as the same than ibrutinib arm. For example, the patients treated with zanubrutinib in ASPEN-study reached 36% of VGPR and CR as compared to 25% in the ibrutinib arm at 44 months. So zanubrutinib is very effective and at least as effective as ibrutinib. And regarding the PFS, the median PFS is not reached in the two arms, but the rate of PFS in the zanubrutinib arm is 78% and at 44 months and versus 69% in the ibrutinib arm.
Although not statistically significant at primary analysis, there was a trend to fast and more durable responses compared with ibrutinib. What does that mean?
I think that it's very important in Waldenström patients who have high tumor burden or active IgM complication to reach a very rapid and deep response. And if we compare the rapidity of the response in the two arms, zanubrutinib led to a very good response rate in 6 months versus 16 months in ibrutinib arm.
How were the responses with zanubrutinib in patients with a CXCR4 mutation, and what does this mean for this specific patient group?
We know that ibrutinib in patients who are CXCR4 mutated is less effective than in the patients who have a CXCR4 wild type gene. And with a longer time to response and also a lower response rate. And in this randomized study, we see that the zanubrutinib is more effective in this population of patients who are CXCR4 mutated with time to major response shorter and also a higher response rate. So I think it's very important because this population represents 30% of the Waldenström patients. So it is the first time that we can see the effectivity of BTK inhibitor in this population.
Can you comment responses to zanubrutinib in patients with MYD88 wild type?
So very few data are available in this population. We have very scarce data with ibrutinib and with a very poor response rate in these populations. And it is the first time that we have a large, not very large, but important populations to answer this question. And we see that zanubrutinib is effective in these populations with 65% of response rate, and also at 44 months, 53% of PFS. However, if we compared in the ASPEN-study patients who are wild type for mid-88 and mutated mid-88, the PFS rate seems to be lower. But it's not possible to compare both, but I think it's not the same populations, but perhaps it's less effective in these populations, but we need more data.
When it comes to safety and adverse events, how compares zanubrutinib with ibrutinib?
If we look at the overall safety, the rates of adverse events leading to treatment discontinuations or to dose reductions are lower in patients treated with zanubrutinib, with 20% of discontinuation of treatment for ibrutinib treated patients versus only 9% in the zanubrutinib arms, and also 26% of dose reductions in the ibrutinib arm versus 16% in the zanubrutinib arm. I think these results are very important regarding the safety profile of this drug, because we know that it's important to keep the treatment, to keep patients under treatment when they are treated with BTK inhibitors.
With longer follow-up, are the safety advantages of zanubrutinib consistent with less off-target activity compared with ibrutinib?
So the kinase selectivity of zanubrutinib explains the less toxicity of this drug, with a less cardiac toxicity, with a very lower rate of atrial fibrillations and also of hypertension. So I think that in elderly populations with cardiovascular comorbidity, I think it's important to have a safer drug to treat these patients. And also, conversely to ibrutinib, there is a higher rate of neutropenia in this trial, but the same rate of infections and grade 3 infection. So I think that the safety profile of zanubrutinib is better than in ibrutinib.
What's the take-home message? Do these results have an impact on the way you treat your current and newly diagnosed Waldenström patients?
So I think regarding all these results, I think that zanubrutinib is safer than ibrutinib, with less cardiac toxicity. I think it's a major complications of BTK inhibitors. And in elderly patients, we know that 20% to 30 patients have cardiovascular comorbidity when we start the treatment. So I think the safety of this drug is a very important progress. And also, we can reach a deeper response, a longer response. And so I think that we, in my opinion, I would choose to treat patients with BTK inhibitor the most safer and the most effective treatment. So I would prefer, in most of the patients treated with BTK inhibitors, to use zanubrutinib in first line or in relapse setting.
Thank you very much for this interview.
You are welcome. Bye-bye.